Okay, so we're going to talk a little bit about translation and transcription and translation. Transcription happens inside the nucleus of the cell. Translation happens in the cytoplasm or the cytosol, whichever, I guess more accurately, it's the cytosol it takes place in. So you have in the nucleus, you have your genetic code, your DNA. That is the double helix of complementary base pairs. We've kind of talked about that before. That, remember, is existing in a unwound form when the cell is just being the cell. And what happens is it splits apart. This is in response to enzymes. Enzymes cause the bonds to break down and to split apart in very specific areas, and there isn't anything about this that's random. Those very specific areas are already coded on the DNA, where you have on the whole DNA line, you have areas of start and stop with regard to these 100,000 proteins that the body can make. And so what you have are enzymes that create these promoters, and then enzymes that create the breaking off of areas where proteins are going to be coded on the DNA. So the DNA unzips, messenger RNA comes in based on enzyme direction. These enzymes, remember, that are made by the DNA itself, so it is directing all of this. And messenger RNA is going to be a mirror image of the DNA and of the complementary base pairs, except for the fact that messenger RNA is going to have uracil instead of thiamine. Sorry, my brain is going crazy here. And so what happens is that messenger RNA will break off based on enzyme activity and will leave the nucleus via the nuclear pores, go to the cytosol, and then and attach itself or associate itself with a free ribosome. That free ribosome, remember, has two units. Part of it is ribosomal unit, the other part is ribosomal RNA, and that's going to attach to that basically lie in between the two units of the free ribosome, and then translation, so tRNA is going to bring amino acid units to that messenger RNA, and those amino acid units are going to be, bonds are going to be formed to make polypeptide chains of proteins. And that basically is how DNA can direct the production of proteins outside of the nucleus without even leaving the nucleus. DNA is it's much too large of a molecule to leave the nucleus. Besides, we don't want it to leave anyway, because you have miles and miles and miles of complementary bases in DNA, and only small portions of them are active to make a particular protein at a particular time. And so you have this concept of transcription and translation. Transcription takes place inside the nucleus. Translation takes place outside of the nucleus. A little bit of information about transport across that cell membrane. Remember that cell membrane? We started talking about that quite a long time ago. It does act as a barrier. It does let some things in, so it has a permeability. And we considered the cell membrane semi-permeable. So some things get in, some things don't. but there are different types of permeability, and actually, to tell you the truth, depending on where you are in the body and what particular cells that you're talking about at what particular time, and I'm specifically thinking of the urinary system, that permeability changes, and it is something that certain cells have the ability to regulate their permeability. So generally we think of the cell membrane as selectively permeable, so allows some things in, not others. But that selective permeability is based on certain characteristics: size, electrical charge, molecular shape, and lipid solubility. So the smaller, the more likely it's going to get into the cell. If in fact it's an electrical charge that is attracted, it'll be more likely to get into the cell as opposed to repelled. Specific shaped molecules will be able to get into the cell via those integral channels, those protein channels that we talked about earlier. And then because the cell membrane itself is lipid based, if something is lipid soluble, it will actually move in to the cell across that lipid, the lipid composition of the cell membrane itself. Transport can be active or passive or active, I guess I should say it that way. Passive doesn't require any energy whatsoever, and that is certain items get across passively, like oxygen gets across. It's a dissolved gas. Active requires ATP, all of that ATP that your mitochondria is busy making all the time. And diffusion is a type of membrane transport, and that can be passive, it can be carrier mediated, which can be active or passive, or it can be vesicular transport. Vesicular transport isn't diffusion. Diffusion is either passive or active, and then with a carrier mediated transport. The thing that helps diffusion is the fact that these molecules or these items that are trying to get in or out of the cell are in constant motion, and that random motion causes them to mix, and that mixing will shift the concentration gradient at that particular point, at that particular area, at that particular point. So diffusion is passive as long as it is the solutes are moving down a concentration gradient. So going from an area of high concentration to low concentration, it's going to be passive. Factors, a few more factors influencing diffusion. Distance. So the farther it has to move, the less likely it will diffuse. If it's right next to the cell membrane and it is something that'll diffuse across the membrane, it'll go in there. Molecular size, smaller, faster, we've done that already. Temperature, the warmer, the more likely, so the warmer the core temperature, more likely it'll diffuse. And then concentration gradient, electrical forces, talked about that already. And then most of these things, if they are in place, diffusion is going to be simple diffusion is what we call it, not carrier mediated diffusion. If it's lipid soluble, like alcohols, fatty acids, steroid, hormones, they will diffuse through because they just become one with the membrane. And then dissolved gases, as I said. With regard to channel mediated, it depends on, still have to be water soluble and ions. And this, the thing that is still going to affect channel mediated is size and charge. So size, particularly the specific shape and charge, you might say, and the interaction with channels. This is a nice picture of diffusion, and you have a little bit of an explanation, a little bit of a image to go with the explanation, and again, this comes straight from your book. But what you can see, what this is showing on this cell, you have a cell and then the extracellular fluid. it's highly concentrated within the cell in terms of this CO2, this carbon dioxide. So that is actually going to move out of the cell until the amount of carbon dioxide is the same outside the cell as inside the cell. And that's going to be a passive process, and that's a good thing because we don't want that carbon dioxide in there. And then facilitated diffusion, again, this can be passive or active. This particular image is showing you a passive one in the fact that glucose actually fits right in that carrier, that channel, that protein carrier. And what happens is it gets down in there based on the shape. And when enough of them get in there, it actually causes that to open up. It's not based on weight or anything, it's just based on the shape of the glucose molecule. Causes that to open up, and that's a passive process. Active sodium-potassium pump, and with this what you have is the use of ATP, and what you're trying to do is pump out, what the cell's trying to do is pump out sodium, so Na plus, out against a concentration gradient. You actually have more sodium on the outside of the cell than inside, but the cell's still trying to get sodium out. It's just the way it operates with regard to the transmembrane potential, so the polarity of the membrane. And what happens is you have sodium being pumped out and potassium allowed in against a concentration gradient. So it does take ATP to get these out. And then you have it all put together, or most of it put together here in a bigger image. Osmosis is a special type of diffusion. Osmosis is diffusion of water across the semi-permeable membrane. And what happens is water will diffuse based on what the solutes are. And those solutes are can be all of the things are in solution, or they can be special, are a particular type of thing in solution. So you have water and then you have the solute. And water is a osmolality, sorry. So the ability for this to happen actually is opposed by the hydrostatic pressure, so the pressure of the water. And the thing that affects osmosis is concentration gradients again, and this opposing pressure of the water against the fact that what you have is, instead of these solutes moving, you have water moving. So water is not going to be able to move against itself, so that opposing pressure of water is keeping water from moving against itself. But this one is happening in the cells all the time. It's a little bit more, I guess a little bit more important. So the water molecules diffuse across the membrane in order toward the solution with more solutes. So what's trying to happen is create a equal concentration gradient of solutes by moving the water. And so volume is going to increase inside the cell or outside the cell. And this becomes very important with regard to individual cells and their ability to live. So you have this concept of tonicity, osmolarity, that was the word I was trying. So you have two fluids have equal osmolarity but can have different tonicity. So they can have equal amount of hydrostatic pressure or solutes, but not the same amount of flow of water in and out. So if it is the same, then it's isotonic. If there are less solutes, it's hypotonic and it's going to lose water. If it's more solutes, it's hypertonic and it's going to gain water. And so in a hypotonic solution, the cell is going to gain water and then rupture. In a hypertonic solution, the cell is going to lose water and shrink. So either way, it doesn't mean good news for the cell. And we will stop there and move on with the next thing in the next part.